Hello, everyone, and welcome to the Phytobiomes Alliance webinar series and our discussion for March of 2023. My name is Dusty Gallagher. I'm the project manager with the Phytobiomes Alliance. We have an excellent discussion for you today. And before we begin, I would just like to provide a little bit of information about the Alliance. So the International Alliance for Phytobiomes Research is a nonprofit consortium of industry, academic, and governmental scientists. We're represented by a number of sponsors that without their contributions, we couldn't do what we do and hold this webinar series. So we appreciate the companies that you see here and the entities you see here on the screen as our Phytobiomes Alliance sponsors. So just briefly, when we talk about the phytobiomes, we're talking about a complex system of plant-based agriculture. So that encompasses everything that you see here, all the way from our microbiomes, microorganisms, our macrofauna, to our substrate, soil, or possibly other types of substrates, plants, arthropods, other animals and plants, and our climate and our weather and our water availability. All of this is influenced by management practices. So we like to consider the complex system in all of what we do at the Phytobiomes Alliance. We have a vision that by 2050, all farmers have the ability to use predictive and prescriptive analytics based on their geography, their biological conditions, and the crops that is the best combination for them producing the food, feed, and fiber for what we need in the globe. And I would just like to announce that our next webinar will be April 26th, and we will be discussing our scientific collaboration in interdisciplinary and international settings. And we have a couple of speakers lined out for you for that next webinar in April. So we'd like to thank you for your participation in our webinar series. And before we begin, want to also just give you a couple of additional dates and some Phytobiomes Alliance workshops. In March 28th, which is just next week, we will be participating in One Health, Phytobiomes, and Animal Science with the British Society of Animal Science Conference in Birmingham, United Kingdom. And then later on this summer in August, we will be at the International Congress of Plant Pathology with a workshop entitled Phytobiomes Research for Plant Health. And then we'll also have a second workshop with the United States Culture Collections Network that will be discussing harnessing culture collections for improved plant health. So be sure to put these dates and these activities on your calendar. Join us if you are able. So just a few announcements before I introduce today's speaker. The webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel, which is listed there for you today. And so if you didn't get a note or would like to review the materials presented today, you can always take a look at that recording. And just a little bit about our platform today. We have 3 different menus that you might be able to access during the discussion. We have the questions tab, which is the question mark, surrounded by a little dialogue bubble. That is where we would like for you to submit your questions for today's discussion. So only questions will go in that Q&A panel. If you would like to monitor the chat, which is simply 3 dots within that dialogue bubble, to see messages from the organizers, And then any handouts will be posted under the handouts tab in PDF so that you're able to download that during the presentation. So again, just a quick reminder for the Q&A session, please use the question panel to submit those questions, which we will have time for after our presentation. So without further ado, I'm very honored to introduce our presenter for today, Rodrigo Alegria, who is a postdoctoral research in AGBS from the University in Morocco, and he's a visiting scientist in sustainable soils and crops from Rothamsted Research in the UK. So I will turn the platform over to Rodrigo for your presentation.
Okay, so thank you very much for the introduction. Can you see the screen well?
Yes,
okay.
Just make sure that you're in presentation mode. And
yeah, okay. Perfect. So let's get to this one. Okay, so yes, thank you very much for the invitation to the Phytobiomes webinar. Very pleased to be here, and okay, just want to get the presentation. Oops. Okay, right. So yes, I'm gonna talk about the research I did while I was in the University of Dundee and based in the James Hutton Institute. So that's part of my PhD and postdoc. So I did that on the Davide Bulgarelli lab. And the title of my talk is A Walk on the Wild Side, the Genetics of Host-microbiota Interactions in the Barley Rhizosphere. So it's titled like that because it's comparative analysis of the microbiotas of wild and elite modern barley varieties. So a brief outline of what I'm going to talk about today. So first, an introduction about concepts some of you might be familiar already, such as the rhizosphere microbiota, and talking a bit about the barley as a crop. And then getting into the research. So we'll try to focus on two questions. So one is related on how the plant adaptation impact the rhizosphere microbiota structure, their taxonomic composition. And also looking at nitrogen, we will see what is also the impact of nitrogen limitation on the rhizosphere microbiota function. So yes, why is all of this important? So the soil is where most of the food production, more than 90% of the food production, directly or indirectly take place. So the soils are full of microbes, like in terms of quantity and in terms of the taxonomic diversity is huge. So the plants and the microbes, they are capable to interact with each other, and also the microbes in the soil, they drive the nutrient cycles. So they are very important. We need to consider them in terms of food production. So if we really want to achieve the full potential of the food production in agriculture and to do it in a sustainable way, we can no longer ignore the microbes in the soil. So we need to deal with them. So the rhizosphere microbiota, it's the rhizosphere first is the interface between the plant, the roots, and the soil. So it's where the plant and water uptake takes place. So the plant release a huge amount of the carbon from photosynthesis actually below ground. So it's really shaping the microbial communities that are around the roots. So they create in particular communities, which are termed the rhizosphere microbiota. So these rhizosphere microbiota, they are capable of performing plant beneficial functions, such as enhanced nutrient cycling or pathogen protection. So if we consider this rhizosphere as an extended part of the plant, so we could thinking about managing it like the same way the upper part of the plants they've been used for breeding, for example. So if we increase our knowledge on how the system work, we could potentially be deployment of beneficial function of these microbes in agriculture. So this rhizosphere microbiota, both the structure and the function, there are several factors that determine them. So we have like the soil factors, we have the ecogeography, we have the soil management, and then also the rhizosphere environment and the host plant through the rhizodeposition, immune system, and so on, and also other factors which probably we don't completely understood nowadays, and also their interactions. So it's a complex systems, and all of these determine what is the rhizosphere microbiota structure and function. So today we are going to focus on the ecogeography of these soils, these fields, and also on the soil management, especially nitrogen fertilization, and how this interacts with the host genotype, with the plant host. So we are using barley as a model crop. So barley is interesting for several things. It's an important cereal cultivated worldwide. In Scotland, for example, it's economically important also in terms of brewing and distilling industries, for example. But interestingly for us, for doing research, we have different things which help us. So we have germplasm collections in terms of mutants, wild relatives, the genomes of several barleys that have been sequenced is used for speed breeding, and also in terms of the genetics, it has much simpler genetics than other cereals. So it's easier to disentangle certain interactions between the genetics and other factors. So it has a diploid genome. Consider, for example, compared with wheat, with some types of wheat, they have an hexaploid genome, for example. So also white barley is very interesting for research. So it's the adapted niche of where the modern cultivar evolved. And also white barley is adapted to more soil marginal areas. So it's potentially developed mechanisms to cope more with lack of nutrients, for example, or adverse conditions. And also, interestingly, is genetically more diverse than the modern cultivar. So there are things within the genetics that has probably useful traits to deal with these more marginal conditions. So now we're going to see how the barley adaptation, how this evolution in these areas actually impacted the rhizosphere microbiota taxonomic composition. So for that, the barley germplasm, the genetic material we have from barley is it's a wild barley collection, which is termed B1K. So this was sampled by the Israeli geographic area. So the sea we see there is the Mediterranean Sea on the map. And then, so these seeds, what we got from them is the seeds. They were sampled on their location, on the natural location. And they are basically 3 ecotypes. So they were clustered according to genetic and phenotypic traits. So these 3 ecotypes are termed north, coast, and desert. And they somehow reflect these environmental gradients there within this geographic area. So we grew them along with elite modern cultivars, Barke, Bowman, Morex, and Steptoe. So this is a selection of 2 and 3 6-row barleys, also used for brewing and for feed livestock. So we see in here the barley ecotypes. They come from very different environmental regions. So, for example, in green we see the north ecotype, which is near the Sea of Galilee. Then we have the coastal ecotypes, which are on the Mediterranean Strip, coast 1, and then coast 2 is more on the hilly area, but also under the influence of the Mediterranean weather. So these 3 ecotypes, they have, let's say, moderate to good, let's say, rainfall per year. Whereas we have the desert ecotypes, desert 1, which is on the Jordan Valley Rift, actually is below the sea level, but they have very low precipitation. And then we have desert 2, which is the orange one here on the southern Negev, which is very dry area. So they are both considered desertic in terms of the annual rainfall. So the questions we try to see is if these ecogeographic factors, first they left a footprint within the barley genome, and also if the barley genome is actually related to the microbiota structure. So basically, it's the adaptation to the plant left a footprint on the genome, the barley genome, and if the barley genome is actually related to the microbiota structure. So we got all the seeds, and what we did in this investigation, so it's a common environment experiment, let's say. So we grew all the seeds from all these different locations and the modern cultivars within the same soil. So the soil is an agricultural soil from near the research fields on the James Hutton Institute. So the soil itself, it's quite typical, let's say, of the Scottish soils, agricultural soils of this area. And it was left without fertilization for at least 3 years. So it was left like without planting or fertilization for at least 3 years. So what we did, we grew the plants on the greenhouse, and then at early stem elongation, we did the sampling of the rhizosphere and the plants. So in here we have the above ground biomass. And then what we could see here is that all these ecogeographic factors, they have an impact on the biomass. So the genotype, it's actually influencing how much the plants can grow, really. So we see that the elite varieties, they clearly outperform the wild ecotypes. So we see the elite here on magenta, and then there is a gradient which goes from north, then we have the coastal ecotypes, and finally we have the smaller ones, which are the desert ecotypes. So this is the above ground biomass. So if we have a look below ground, so we calculated the root to shoot ratio to see what is the proportion of it's allocated to the roots compared to the upper part. And we see the different trend in which the elite varieties, they are actually much smaller than the wild barleys. And then within the wild barleys, we see the similar trend. We see the north and the coastal ecotypes clearly outperforming the desert ecotypes, especially desert 1 in this case. So yes, the wild barleys, they seem to invest more resources below ground, probably for this adaptation to marginal conditions, natural conditions. Okay. So then we would like have a look to the genome of the plant. So we created these genotypic profiles So we did that with single nucleotide polymorphisms. So it's a SNP chip which contains like about 1,500 SNPs, so single nucleotide polymorphism. And then we created a genetic distance with that. And so we could see on this hierarchical clustering, it's a clear differentiation between the elite varieties and the wild varieties. So that's quite marked. And then also within the wild ecotypes, we see two big groups. So one it would be the coastal ones, the Mediterranean ones, and then the other ones, let's say the more desertic ones, they kind of cluster together. So we see a clear separation on this on the genotypic profiles. So we were wondering if that would be reflected also on the microbiota structure. So for that, we got the rhizosphere from all the plants. So we try to see the rhizosphere microbiome taxonomic composition. So we extracted the DNA from the rhizosphere and the bulk soil. We created 16 ribosomal RNA gene libraries, did Illumina sequencing, data analysis. So then we are able to see with this marker gene for phylogenetics, which is the 16S gene. So we could see like who is there and which proportion the bacterial taxa are there. Right, so if we put the results of this microbial profile from the 16S amplicon sequencing, and we put them in an ordination. So basically in the ordination, so this is with a distance metric calculated. So we could see that the closer the points are, it means they have a more similar microbiota taxonomic composition. So the first thing we could tell, so we have the brownish ones are the bulk soil, and then we have the orange ones, both triangles and circles are desert ecotypes. Then we have the coastal ecotypes, also with triangles and circles in blue. We have the north one in green, and we have the elite varieties on magenta color. So we see first a clear difference between the rhizosphere profiles and the bulk soil. So that's good, that's what we would be expecting. And then we see two major groups, which are more different from each others, which one would be the elite modern varieties, microbial profiles, from the desert ecotypes, microbial profiles. So a bit of somehow what we see within the genotypic profiles of the plant. And then we see this ecotype effect actually accounts for 17% of the total variance within these microbial profiles. So that sounds good. And then so we wanted to relate the microbial profile taxonomic compositions, the microbiota structure, with the genotypic profiles. So what we have on the x-axis is the genetic distance of the host, and then what we have on the y-axis is the microbial distance, so this similarity metric. So we see there is a significant correlation between both. So that's probably telling us that a part of the genome of the plant is actually interacting or shaping, modulating the rhizosphere microbiome. So next we will move to the next part of the presentation. So we had a look of how the plant adaptation shapes the microbiota structure. So now we are going to look a bit more into the function of it, and especially we are going to have a look to nitrogen. So basically it's how the nitrogen limitations shape the microbiota function. So first we will start to see how actual different levels of nitrogen impact microbiota recruitment for the plant, to see if different nitrogen treatments would have an impact on the microbiota structure. So this is just a brief introduction of why nitrogen is a major plant nutrient. So of course it's very important. So plants, they need nitrogen to grow well. And the soil available nitrogen, it comes either from biological fixation for the degradation of organic matter already present in the soil, or by fertilization. So the plants are very good in utilizing the nitrogen once they took it from the soil, but especially cereals, they are not very efficient in uptaking this nitrogen. So many of this nitrogen, they get lost, and it creates nitrogen pollution into the environment. So we were thinking if the wild barleys, they would have microbes, they would be able to be more efficient in that terms. And also because nitrogen has such an impact of the plant, we wanted to see how nitrogen would actually impact the interactions of the host and the microbes. So for that, for this part of the experiment, we moved from the 20 wild barley genotypes we had, and we selected 2 of them, so one from north and one from desert, and also we select an elite variety, which is Morex. So we did that in terms of their contrasting, the most contrasting microbiotas. So we grew the plants in a soil. So this soil was amended with a nutrient solution. And the nutrient solution, it was kept at full nitrogen, 100% of nitrogen, at 25% of nitrogen, and at 0 nitrogen, while keeping the rest of the nutrients to an optimal level. And then so growing these 3 different genotypes, the desert, the north, and the lead. So first we are going to see how the plants grow in all these different treatments. So if we look at the plant biomass on the top figure, we could see that basically speaking, like the more nitrogen you put into the plants, so the more the plants grow. So nitrogen 100 outperform clearly nitrogen 25 and nitrogen 0. Also, we measure the plant nitrogen content. So the amount of nitrogen within the plants is also greater at higher nitrogen concentrations of the treatment compared to nitrogen 0. Also, we had a look about at the time of sampling, we also sampled the rhizosphere, and we extracted the ammonium and nitrate from the rhizosphere. And we see that, again, especially at nitrogen 100, the amounts of ammonia and nitrate, they are much greater at higher nitrogen concentrations. So basically, we could tell like our nitrogen treatments, they do impact the plant biomass and the soil. So basically, our treatment worked. Okay, so we generated the microbial profiles of these different nitrogen treatments and the genotypes. So we could see a separation between the bulk soil and the rhizospheres. So that's again, as we see in previous graphs, so what would we be expecting? And then also we see a separation according to the treatment. So nitrogen 100, which are in triangles. Then we see the round ones is nitrogen 25, and the square ones, which are nitrogen 0. So we see a clear differentiation of the treatment on the rhizosphere microbiota structure. So also interestingly, we see that a differentiation of the plant genotype. So we see how the nitrogen zero, how the desert, the modern and the north, they cluster together. Whereas this differentiation of genotypes, they seem to vanish at higher nitrogen concentration. So it seems like the host control, the plant host genotype control of the microbiota, it's greater under nitrogen limiting conditions. So then we did a statistical test to see which bacteria they would be differentially enriched in pairwise comparisons. So in here we see on the horizontal blue bars, so this is the total enriched in the pairwise comparisons. So for example, here it would be nitrogen zero, the square ones, the one in magenta with the square in orange with this. So there's a comparison between desert and modern rhizosphere profile. So we could tell how many bacteria is differentially enriched. And then we see on the vertical bars, we see the specificity of this enrichment in terms of unique bacterial taxa. So what we could see is like both of these, the total enrichment and the unique enrichment are much higher at nitrogen zero concentrations, whereas it's progressively diminished until it's virtually vanished at nitrogen 100 treatment. So yes, so we could say that nitrogen limitation amplified the host selection on bacterial recruitment. So once we see that the plants that recruit a more specific microbiota at nitrogen zero, we wanted to see how the differences between the genotypes are. So for that, we want to study the metabolic potential of the rhizosphere microbiota. So we're going to focus on the nitrogen zero as we're talking about. So we're going to see this metabolic potential by shotgun metagenomics. So basically, this is the pipeline that we followed. So we started at the same point, so with the DNA, and this DNA just followed a pipeline for metagenomics, so it's fragmented. Then these contigs are assembled. Then there's annotated, there's functional enrichment. We are able to retrieve the gene ontology terms, the terms. And also by reconstructing genomes within this metagenome, we are able to retrieve the metagenome assembled genomes. So in here we talk about entire genomes as opposite with the 16S in which we were using only one marker gene. So we have a look at these metagenomic profiles. So we can see here on the left-hand side, this is the differences between the abundance in reads between bacteria and fungi. And we see that bacteria outperform clearly fungi in our metagenome data. So, and then looking at bacteria, we could see also a clear differentiation of the profiles from the bulk soil to the rhizospheres. And then within the rhizospheres, we could see a cluster of the desert wild ecotypes compared to other one, which is mostly related to the elite metagenomic profiles. So our first question was how this the genes they are actually on the nitrogen biogeochemical cycle would actually differentiate the different metagenomic profiles. So we see the amount of reads. In here, they are only presented the ones they differentiate in the bulk soil and the rhizospheres. So we have several things, genes related to nitrification, denitrification, degradation of nitrogen compounds. But we were okay. So, but there is no significant differences between the genotypes, the different metabolic profiles from the different metagenomic profiles from the genotypes. So we're like, okay, that's interesting. So so then we will have a look to the whole of the metagenomic profiles, and then we could see a difference also between the genotype effect differentiating the elite varieties from the wild barleys. So there's a clear genotype effect here. And then we see also there is a microhabitat effect differentiating the bulk soil from the rhizosphere profiles. So if we have a look to the Genome terms, they actually underpin this ordination. We could see there are several Gosling terms that differentiate between the bulk and the rhizosphere. So for example, in the rhizosphere, we have things like cell addition, pathogenesis, carbohydrate metabolic processing, responses to chemical, flagellum motility. Whereas in the bulk soil, we have enriched for functions related to sporulation, photosynthesis, antibiotic synthesis. So we could see, we could think that these are functions that are related to the bacteria adapting to a nutrient stress in a carbon-poor environment, such as the bulk soil, compared to the rhizosphere. And also we see that several functions they are pointing to these different genotype effects. So we see that elite and wild, they have different functions associated to them. So we could even think that the elite enrich for more functions related to host and microbe interactions, whereas the wild, they seem to be enriching more for functions related to acquiring carbon and carbon metabolism. So then we reconstructed the genomes of several bacteria from this metagenome. So they are termed MAGs, which is metagenome assembled genomes. So so basically this could tell us which bacteria is doing which function. So we were able to retrieve 67 metagenomes from 14 different bacterial classes. And then in here we could see the functions, the GO terms. They are the top GO terms, which actually enriching in terms of function for a particular barley genome. So specifically, we were able, through intersecting the different data sets, a function carbohydrate metabolic process, which is really enriched in bacteroidetes, which is also enriched in the lead genotype. So this is potentially useful in terms, for example, of isolating bacteria. So we could see like this genotype would select for bacteroidetes would put enriched in carbohydrate metabolic processes. Okay. So after all this sequencing data, we wanted to see how it would be the impact of the microbiota on the actual barley growth. So for that, we did a plant soil feedback experiment. So basically, we have the same soil as previously. We use the nitrogen zero solution, and we grew the elite modern variety, and also on parallel the desert genotype. So basically, we grew them from one generation. So the bacteria these soils, they become conditioned because of their host genotype. And then, so with these conditioned soils, we leave them as native condition, and the other one is the one we use heat treatment. So we have elite conditioned soil heat treated, desert conditioned soil, native we call it, and then we have desert conditioned soil, which is through heat treatment. And then we grew the modern variety in all of them, and trying to see if there's differences in the plant growth. Okay, so we could see there are differences on the plant growth when we compare the native soils with the heat-treated soils. So basically, it seems like a functional microbiota is required for optimum barley growth. So this was only significant in terms of the desert condition soil, but we could see differences in both of them. Okay, so if we extracted the DNA from the rhizosphere of these plants, and we generated the 16S amplicon sequencing, so we could see here there is some, we put them into the ordination, and we see this a clear difference between the bulk soil, which are on the right, black and white, with the rhizospheres. But also we see that the heat-treated ones, they are much more alike than each other, where the native ones, they cluster also differentially. So, and then if we do a statistical test for the differential abundance, and we compare the elite conditioned or the desert conditioned native against their bulk soils and the heat treated counterparts, so we see the bacteria that are specifically recruited by them. So, and we could see there are several bacteria taxa belonging to the normal phyla we see on the rhizosphere, and we see they are kind of very similar between each other. So it seems like we grew them in the Morex. So the Morex genotype is actually able to recruit its own microbiota, is converging. So there is a genotype effect which somehow mostly override this conditioning of the soils. So now a bit of summary of all the things we talk about. So the first part of the presentation, we saw like how the plant adaptation shapes the phenotype of the plant, so how these ecogeographic factors shape the above ground biomass and below ground biomass, and how it's also impacting on the genome. But also we see that this adaptation of the plant, the genotype, it's also influencing the associated rhizosphere microbiota structure. Then we saw that nitrogen limitation amplifies the barley genetic control of the rhizosphere microbiota. So it was very clear on the different nitrogen treatments. And then we see, looking at the function, we see how there were very big difference between the bulk soil and the rhizosphere. Within the rhizosphere, a bit more subtle effect of the barley genotype, but so they recruit different functions of their rhizosphere microbiota. And then finally, we saw that a host-dependent microbiota is actually required for optimal barley growth under nitrogen-limiting conditions. So looking a bit to future research and the perspective of it. So we saw that plant adaptation shapes the plant genome, which in turn it modulates the taxonomic composition and function of the rhizosphere microbiota under low nitrogen conditions. So potentially we could manage the rhizosphere microbiota for crop beneficial functions through managing the plant genotype. So to make a more sustainable agriculture. So for example, through breeding, we could potentially be able to modulate the microbiota for plant beneficial functions. Okay, so so that's pretty much the end of the talk. And now I'm just briefly introduce my current job. So my current project. So it's a bit of change of scenery. So now I'm hired by Mohamed VI Polytechnic University in Morocco. I'm funded by OCP, and I'm based in Rothamsted Research, the same project also with Cranfield University. So I'm still looking at the microbiome of, in this case, is wheat and maize ecosystems in Morocco. So we have all the samples, they coming from different places in Morocco. And I'm using culture-dependent and also culture-independent analysis of these microbiomes. So thank you very much for your attention. And also thank you for all the authors and all the people who contributed and helped with this research, especially to Senga Robertson, which is co-author with the nitrogen paper, Joan Russell for her help with the barley genotyping, Yael Friedman for the seeds of the barley ecotypes, Matthew Barrett for the MAX analysis, James Abbott for data analysis, especially on the metagenomic part, and to Davide Bulgarelli, which is the PI of the project. So in terms of funding, I was funded by the Scottish Food Security Alliance through my PhD, and then also after we used to thank the Horizon 2020 program of the European Union, especially with Project Circles, and also to the University of Dundee Department of Plant Science, which is where the research took place. So thank you very much.
Thank you very much, Rodrigo, for a very interesting presentation. We would like to introduce David Bulgarelli, who has joined our webinar as a panelist and one of Rodrigo's colleagues. So he will be available through the Q&A as well. And so hello, David, nice to see you. So as we prepare for the Q&A, I invite everyone to submit your questions to the questions panel. And while those are coming in, we're just going to get started. And actually, I'm going to take liberty and take the first question, if you don't mind, Rodrigo, and just ask, if I understand you correctly, you used the same soil type throughout all of the experimentation. Do you have plans to perhaps look at different soil types, or maybe take the experiments to the field in the natural environment to see what type of interactions that you receive?
Mm-hmm. Yeah. So yes, we use the same soil throughout the experiments. So of course for consistency. We did another different soil also from Scotland at the beginning of the yes, let's see, regarding the first part of the presentation, the plant adaptation part, and we saw a very similar pattern with the desert ecotypes separating clearly from the modern ones. And then, so on the next project I was working in circles, we did sampling on the fields. I mean, we were working, so we moved from the greenhouse towards the field, although this was for spinach and tomato. And on the project I'm working now, also there's been field sampling of the rhizospheres of wheat and maize. So it seems like the research of the rhizosphere is moving more towards the field conditions.
Okay, thank you. So we do have several questions coming in. So our first question is, how many microbiota samples were taken per group between the desert, the north, the elite, etc., those germplasm types?
Okay, so I think that's probably relating to the second part, to the nitrogen treatments. So I think we have, like, depending on the experiment, but for example, for the metagenomic, I think we have 4 of each.
4 of each? Okay.
If I remember, yeah, at least 3 or 4. I think there were 4 of each.
Okay.
Yeah.
And another sampling related question is, how close to the root does the sample need to be taken to be considered as a rhizosphere microbiota sample?
Yeah, I mean, the rhizosphere, I mean, the plant and the roots influence through the soil is somehow a gradient, let's say, coming from the rhizoplane of the root towards the soil. So, I mean, there is not a right or wrong answer in terms of sampling, but if you want to have like more of a genotype effect, I think you would need to get closer to the root as possible. So yeah, in our sampling the rhizosphere, it was like, yeah, very much the soil really attached to the root surface. So yeah, with this lodge, the soil was not really attached to the roots, and then they were like centrifuged several times and so on. So basically, you end up with the roots virtually clean, and then so, but it's, yeah, it's the thin layer through the root.
Okay, our next question is, are you aware of any plant breeding programs that focus on leveraging beneficial plant-microbe interactions, or is this just a proposed breeding scheme?
Okay, I think there is a lot of interest through that. I mean, the things I've seen, I think, is mostly on the research field. So I'm not too sure if there are breeding companies actually looking at it in that way. I don't know if David is aware of it.
Yeah, I may chip in on this one. I would say yes. I can't really disclose details because they are not being disclosed to me. But yeah, I think this is becoming a reality. The nature, the form, and the scope. This is again still to be fully defined, but it's not just a nice idea. It's becoming to become something translated into practical applications.
Okay, thank you. Our next question has to do with the cumulative reads. And the question is, the cumulative read seemed quite different between the bulk and the rhizosphere samples. Is this related to total microbial biomass or sequencing depth?
Yeah, which slide do you think it's related to? So probably it's this one. Okay. So in here they are only shown Yes, okay, so this is the rhizospheres, yes, and the bulk soil. So yeah, there is always differences between the rhizosphere and the bulk soil. And what is interesting here is like the heat-treated samples. Also, they seem to lack this bacterial microbiota, which seems to giving the extra bit. So in terms of plant growth, so they seem to be necessary to achieve optimum growth.
Yeah,
so they are meant to be different, I think.
Yeah, so if I can add just one comment on that. So it's not just a difference of sequencing depth. So here the data where, let's say, the input data is the same here in terms of sequencing depth. You see this marked difference because those were taxa significantly enriched in the rhizosphere compared to the other samples. It means that the other samples may have other bacteria that are less abundant in the rhizosphere, and those are the ones that are, let's say, predominant in the rhizosphere samples and were significantly enriched there. It's not just that we have sequenced more rhizosphere than bulk. So the input, the sequencing depth is the same. It's just to reflect the differential enrichment.
Okay, have you compared the rhizosphere microbiotas of the different ecotype barleys using soils from those ecotype regions?
Yes, we were thinking about that for a while, but it's thing is we would need to ship soils from Israel to Europe, and then that was not that practical. I mean, in terms of practicality, it's not so easy to get permission and to get the soils, but we were thinking that would be a very interesting experiment.
Okay, is there any indication that the desert ecotype microbiomes might buffer plants against drought stress?
Yes, I mean, we didn't look for that, but I think it's a very interesting hypothesis. So we focused on nitrogen and then the taxonomy differences and these ecological patterns. But I think these ecotypes, they are mostly defined by rainfall. So it seems like and very interesting things to look at.
Okay, and I think we have time for one last question today. So what is the rationale behind the heat treatments of the soil, and what was the temperature of that heat treatment?
So the soils, they went through the autoclave. Yeah, I mean, I don't recall the exact program of the autoclaving. So they were like, yeah, most of the things, they were, you know, bacteria, they were basically killed, most of it. You cannot really tell it's totally sterile because, yes, that's a bit more difficult. Then we have a look to how the chemistry of the soil changes. And then there are changes, but these changes, they don't point to any particular genotype. So the treatment has an effect on the soil, but this treatment is not biased, let's say, towards any of the genotypes.
Okay, thank you very much. And so our time is drawing to a close here today, and so I want to give...
I can add on the heat treatment. I just went to the paper so to look through. It's 126 degrees for 1 hour, repeated twice at an interval of 12 hours. So this is the heat treatment that we have performed. And it's written also in the manuscript, so there will be also additional details on that. And if I may add on the rationale, so what we wanted to do is really basically disrupt what is the endogenous microbiota, because we knew that this treatment would have affected, even if not, let's say, sterilize or wipe out entire communities, would have affected in a way that the plants were not exposed to what we thought was the endogenous microbiota. And then our data are supporting that.
Mm-hmm. Okay, thank you for that clarification. So once again, Rodrigo, thank you for an excellent presentation. David, thank you for joining us in this Q&A session. And I want to thank all of our attendees today. And please join us for our April 26th webinar, Scientific Collaborations in Interdisciplinary and International Settings. So I hope everyone has a great day. Thank you for joining.